Of course, is an immunosuppressive drug. It's a drug that allows you to do kidney transplants and heart transplants and things like that. And it wipes out the activity of this peak. Why does it go up? Well, there's no calcium in the unactivated egg. Add calcium, whoosh, up goes the activity. The calcium is then sucked out into those little vesicles, so the activity of the enzyme goes down. And you can wipe it out with cyclosporin A. Great. So what's funny about this is that nobody ever thought about this. And actually, if you think about it, you might be a bit puzzled that I've told you that a protein kinase is turned on by the calcium. So it seems a bit funny to turn on both a protein kinase and a protein phosphatase at the same time, because in principle, nothing should happen. But actually, the substrates of the two things are different. We still don't actually know what the salient substrate of the calcineurin is. So the next question was, does the cyclosporin work? The answer is yes, it does. And what's the consequences? Well, this is a typical example of how experiments in this system work. Here's the destruction of cyclin B in response to calcium. And when you inhibit the phosphatase, you can see it's sort of trying to degrade the cyclin, but not doing a very good job of it, although the cyclosporin A keeps the APC3 up high for a very long time. So this was a sort of typical, you hope for a really clear answer and a really clear effect, and it wasn't totally clear. So we did some more experiments, and we made the experiments more physiological, if you like. And we did that by adding nuclei, sperm nuclei, to these extracts. Now, what should happen if you add sperm nuclei and you add calcium and there are no inhibitors is the chromatin in the sperm nuclei swells. A beautiful nuclear envelope is formed around the chromatin. The DNA replicates, and this nucleus will suck up nuclear proteins, fluorescent nuclear proteins, which we've added in here, a GFP, nuclear localization signal. And this assay allows you to quantitate, because if you add cyclosporin A, what happens is the chromatin does not swell, the nuclei do not reform, and of course, because there are no nuclei, they can't suck up nuclear proteins. And this allows you to quantitate the timing. And amazingly, if you inhibit calcineurin with cyclosporin A, you induce a 1-hour delay in the return to interphase. So the activity of this enzyme, this phosphatase, is really important for getting back into interphase. So we thought, well, gosh, it's time that we really started assaying phosphatases, and we had a very lucky break here because Satoru made a synthetic substrate. It's very important when you're assaying phosphatases to only have one phosphate in the substrate, because there are an awful lot of phosphatases, and if you have more than one phosphate, the chances are that a different phosphatase will dephosphorylate the neighboring one. So you make a simple substrate, and this was a simple substrate, 25 residues from a well-known mitotic phosphorylation site, fused to maltose binding protein, so you can make it in bacteria. Purify this protein, phosphorylate it in vitro with cyclin A CDK2, which we have in the freezer, and gamma P32 ATP. You make this labeled substrate, you add it into the extract, and then you take out samples. Usually what happens is you add it in and then take out a sample about 2 minutes later, so we measure the instantaneous rate of dephosphorylation. And we got a big surprise here. So this is what the data looked like, and when we first saw that, we thought, oh, well, this is just like an undergraduate experiment where he dropped the tube at that point. But this was Satoru, and he doesn't drop tubes. And this is a very reproducible result. And the answer came very quickly when you add inhibitors. So if you add okadaic acid, which is a well-known inhibitor in this system at the concentration we use of both PP1 and PP2A, you have no effect on the first peak, because that's the calcium-activated phosphatase, but you wipe out the second peak. And of course, if you add cyclosporin A, you wipe out the first peak and delay the appearance of the second peak. So what it is that actually, to get out of mitosis, to dephosphorylate all those substrates in this particular situation, when the sperm arrives, you need not one phosphatase, but at least 2 phosphatases. And no wonder we were a little bit confused. And there's this spike of calcineurin which somehow unlocks the appearance of the second phosphatase, maybe just by degrading the cyclins, we don't know. So we called the second phosphatase phosphatase X for obvious reasons. We had no idea who it was. And the only experiment which we could do was this, to assay phosphatase X. And we were absolutely delighted when my prediction from the plenary lecture turned out to be absolutely correct, namely phosphatase X, as measured by that particular substrate, don't forget, completely turns off when these extracts, these Murray extracts, enter mitosis and come back on again when they come out the other side. Great. Absolutely great. So this explains why we don't get futile cycles. But it doesn't help us understand what phosphatase X is, whether it's one thing or lots of things. So two obvious questions. What is it and how is it turned on and off? And I'm going to tell you the answer, because we now know. I didn't think we would know by now, but we do. So the most likely candidate from lots of experiments, which I won't go into, mostly due to using this inhibitor okadaic acid, is that it belongs to this family of protein phosphatases, but there are quite a lot of catalytic subunits. And the main ones are PP1 and PP2A. Where's PP2A? There, up at the top. And they all belong to the same family. So what Satoru did was to simply try and immunodeplete. You can't make knockout frogs yet. So what you can do is take these very crude extracts, add antibodies, and suck out the relevant protein. So he made, excuse me, I always never quite know where to show this slide. I mean, originally we thought, oh, we'll purify the phosphatase that turns on and off. Okay, and that'll tell us who it is. So you do this experiment. This is a very puzzling experiment. So in order to purify the phosphatase and identify it, you, of course, have to make extracts, and you then dilute the extracts in the course of purification. And what Satoru found, the amazing thing, so here is a mitotic extract, no activity whatsoever against this substrate. There's the interphase, 100 times more activity, right? But when he diluted, he just added buffer. I mean, ordinary tris buffer with magnesium, where it contained ATP and all those kind of things. Amazingly, there's a huge activation just by the dilution. And we asked phosphatase experts about this, and they said, oh, yeah, it's well known that when you dilute the extract, you get more phosphatase. This is how you get lots of phosphatase. But, of course, that meant that we couldn't identify it anymore because we lost the difference between interphase and mitosis by doing this. So that was, and we couldn't understand it, what we were doing by the dilution. We'll come back to that. So we had to, what Satoru did was just to clone all of these catalytic subunits, and it turned out that the catalytic subunit of PP2A and PP4 didn't work very well, so a friend provided a monoclonal antibody against a different subunit. And I'd like to draw your attention to the concentrations. Much the most abundant one is PP2A. I mean, it's 8 times as abundant as PP1, right? But, there are quite a lot of them are present at some concentration or another. And it turned out that the depletion of PP2A depleted, and that was sort of what we expected, really. And that's fine and well and good, but unfortunately there are a lot of different forms of PP2A. I don't know if any of you have followed this. They have terrible nomenclature. Phil Cohen invented this nomenclature. They are called B, B prime, B double prime, and B triple prime, and I challenge anyone to search PubMed with B triple prime. And then they have, sometimes they're called alpha, beta, gamma, delta, sometimes they're called PR74, PR4. I can't tell you whether PR48 is an alternatively spliced form of PR74 or whether it's something completely